So hello, everybody. My name is Anna Mearson, and I'm the Vice President of Commercial Services at Biologics Consulting. And today I'd like to welcome you to Breaking Through the NAMS Noise, the impact of FDA's roadmap on NAMS or new approach methodologies for drug safety testing. I will be the moderator for today's webcast, which is sponsored by Biologics Consulting. Biologics Consulting is a full-service, end-to-end regulatory consulting organization, and we're focused on providing FDA approval, product development, and regulatory consulting support to sponsors that are developing complex drugs and biologics, combination products, and medical devices. With a 30-year history, we lead with strategy and science while leveraging deep expertise and understanding of the regulatory pathways for optimizing product development for our clients. Our speakers today are Dr. David Pepperl and Dr. Bruce Pearce, and I'll briefly introduce them now. Dr. Pepperl is a pharmacologist and toxicologist with experience in early stage biologic development. He has been with Biologics Consulting for over 20 years and provides thought leadership as a principal non-clinical consultant, advising clients seeking regulatory guidance with their preclinical safety toxicology studies. Dr. Pepperl comes to us from Theramun Research Corporation, where he served as a toxicologist and preclinical development scientist. And Dr. Pepperl's expertise lies in design and development of numerous non-clinical development programs for a variety of biologic products, including cell and gene therapies, mRNA therapies, vaccines, blood products, monoclonal antibodies, live biotherapeutics, protein therapeutics, and small molecules. Dr. Pepperl holds a PhD in pharmacology and toxicology from the University of Arizona, and he subsequently completed postdoctoral work at Pharmacia and Parke-Davis Pharmaceuticals, with a focus in vitro and cell biological models of drug action safety and signaling. Next, we have Dr. Bruce Pearce. Dr. Pearce is a classically trained pharmacologist and toxicologist with broad expertise in early and late stage development of small molecules and biologics. He has spent the last 15 years of his career as a principal non-clinical consultant at Biologics Consulting. Prior to his tenure here, he held a number of senior level positions in both the biotech industry and academia, including as a director of clinical research and pharmacology, as a trauma program manager for a hemoglobin-based blood substitute, and as a principal in the development of a botulinum toxin-based therapeutic. Dr. Pearce's domain expertise comprises design, oversight, and reporting of in vitro and vivo pharmacology studies and safety pharmacology, toxicological testing of small molecules, biologics, and combination products, developmental and reproductive toxicology testing, and toxicological risk assessments of drug candidates and excipients. Dr. Pearce completed his PhD in pharmacology and experimental therapeutics at the State University of New York at Buffalo and was an NIH postdoctoral fellow at Yale University School of Medicine and Harvard Medical School. He has more than 85 publications across peer-reviewed journals. And with that, I'd like to turn it over to David to get us started.
Thanks, Anna, and I appreciate the opportunity to speak today. So we'll be talking about NAMS and breaking through the noise and what this all is about. So I'm sure most folks have heard about NAMS in the course of 2025. There's been a lot of talk and buzz through the year. But what exactly are NAMS? So just a quick definition that's out there. Certainly, NAMS are simply novel methods, scientifically fit, valid methods, leveraging non-animal testing strategies such as organoids, AI machine learning, cell-based assays, which are intended to replace, reduce, and refine animal testing in the spirit of the 3 R's for drug product or safety testing. So NAMS generally fall into several buckets, and certainly AI machine learning is one, but there are a number of kind of general buckets that these fall into. The first is microphysiological systems, which tend to be interconnected systems of 2D or 3D human cellular constructs where the drugs can be tested against the human cellular models. Whereas complex in vitro models are a little more complicated, but typically involve 2D and 3D cultures supported by tissue-derived matrices containing immune or possibly other supporting cells. And finally, another approach that really doesn't involve any animal or experimental testing is weight of evidence approaches, which are essentially systematic approaches for decision-making, integrating multiple sources of data. And Bruce will talk a bit more about these as we move forward. advance here. Okay, so we've seen this explosion of NAMS talk and efforts in the course of 2025, but why this sudden push for for development and use of NAMS? Well, certainly there have been a number of changes at the U.S. FDA with the new administration pushing for improved methods to speed drug development, make drug development more efficient. There's also quite an effort right now. Oh, hang on a second. Went forward to back up a slide here. Okay. Certainly, we've been finding that animal studies are often inadequate to accurately predict human outcomes, such as the T. Gennaro incident, where the drug was found to be safe, but then caused a severe reaction in the phase 1 volunteers. So that's certainly a big driver of this all. And products are just simply much more complex and specific than they used to be. Most products, including monoclonal antibodies or human cells and tissues, can't adequately be tested in animal models. So there's been a need for increasing use of humanized or human models for drug safety testing. And that has led to, certainly in parallel with a rise in the technological advances that we've seen in cellular and organoid technology, AI, ML approaches, and drug safety testing. So, and ultimately, the desire is to decrease animal use and certainly cost for developing these programs. As we've seen the cost of, for instance, non-human primates simply explode over the past few years since the pandemic. So this use of NAMS would help bring down the cost, make more models, our approaches certainly more humanized. Next slide. So NAMS have really been around for a long time. The name is relatively new, but I'll get into a little bit of the history in a moment. But certainly, we have things like in vitro efficacy assays that have been around for decades, ligand binding assays, cell-based functional assays, as well as assays we currently use for GLP safety testing that don't involve animals at all, including Ames assay and bacterial mutagenesis, or tissue cross-reactivity for monoclonal antibodies to test whether an antibody will cross-react with human tissue, with the human tissues, or with specific animal tissues, as well as other assays which have been used historically in other industries, particularly in the environmental and chemical industries, in vitro skin sensitization assays, as well as ocular irritation assays, the bovine corneal opacity test, for instance, to look at eye irritation. These have been used for years and have been approved and accepted by regulatory authorities. And that leads me into sort of a little bit of brief history of where this all came from and what the impetus is for leveraging these NAMs. So it really came out of the Environmental Protection Agency years ago requesting a study by the National Academy of Sciences on toxicity testing in the 21st century, citing the need for new approaches in chemical safety testing and to provide a summary on new approaches that can be taken to streamline drug safety. And at the same time, in about the year 2000, ICVAM, or the Interagency Coordinating Committee on Validation of Alternative Methods, was established. And for the first 15 years or so, a lot of this was in the hand of researchers and NIH scientists, and a linear stepwise approach had been used with NAMs without much dialogue between the NAM developers and the NAM end users. So what was really needed was less emphasis on direct, immediate use of these NAMs as replacement therapies, and more emphasis on the NAMs that can be incorporated or incorporate batteries of NAMs to address key specific questions. And And industry also determined that it really realized that it needed more dialogue between developers of these NAMs and the end users. So that in 2018, after all this time, ICVAM developed the strategic roadmap, essentially a resource for federal agencies and product developers to seek and develop new approaches to develop NAMs to improve risk assessment and improve human relevance and ultimately decrease animal use. So things started moving pretty quickly. And in 2022, the FDA or the Congress passed the FDA Modernization Act 2.0. And this was signed by President Biden, leading to a flurry of activity because this act stated essentially that animal studies were no longer required for product development. Well, this was partially true, but what it really said was it allowed for these use of these alternative approaches or NAMs, alternative assays that will help decrease animal use and increase specificity of the data toward humans. So over the next few years, things started to move along, develop. And in 2024, the ICVAM published a document entitled Validation, Qualification, and Regulatory Acceptance of New Approach Methodologies. And this is cited in my references at the end, but basically a guidebook as to how to validate and develop NAMs. And I'm sure most of you are aware now that early last year, April of last year, FDA released its guidance document on FDA's roadmap as to how to reduce animal testing in preclinical safety studies, termed the roadmap. And then just recently in December of last year, FDA released its guidance document on alternative approaches and strategies for non-clinical testing of monoclonal antibodies. Okay, so currently what we have now in 2026, these are just a few examples of some NAMs. We have in vitro humanoids, human systems with organoids and spheroids, microphysiological systems that I mentioned before, in silico and computational models, physiologically based pharmacokinetic modeling, PK, AI, ML predictive modeling. And then again, weight of evidence approaches are a kind of a fixture here and are widely used in carcinogenicity and developmental reproductive toxicology safety assessments. And Bruce will talk a bit more about those approaches. And certainly these NAMs have been used quite frequently and are being adopted by the contract research industry, leveraged for, there we go, for use. And certainly a number of contract labs are leveraging these models, organ and tissue models, 2D and 3D, iPSC-derived organoids with human-based cells, human microarrays using ion channels and transporters are under significant development, human protein microarrays, as well as digital and virtual tools that we've been seeing and hearing a lot about, including digital twins of organs and test systems that have been widely used across numerous industries, virtual control groups, which can save animals in studies where there are historical databases. And then again, AI machine learning for drug discovery, and those are being extensively all used today. Okay, so these are all great and well, but these methods mean nothing if they're not accepted by the regulatory authorities. And FDA in their Modernization Act essentially allows for alternatives to animal testing, which include NAMs and other approaches that will address the key questions that must be addressed to go into first-in-man trials. And that's all great. However, FDA still requires sufficient supporting efficacy and safety data. And that's just something we have to address no matter what methods we use. And FDA has long supported these use of in vitro or other alternative approaches to decrease animal use. You'll see in their FDA comments, typically from INDs and pre-INDs, where they suggests that alternative methods can be used in lieu of animal testing. And in fact, NAMs are also used quite extensively in regulatory guidelines. If you go to any of these guidelines published recently, even since 2005, photo safety, reproductive developmental toxicity, FDA guidance on immunotoxicity, or QT cardiac safety. There's a number of NAMs that have been listed there and have been widely used and promoted. Neutral red uptake for photo safety, human skin models, other reproductive models, mouse embryo, stem cell safety tests. And certainly for immunopharmacology and immunotoxicity, there are a number of methods that can be used in vitro using human cells, cytokine release assays, TDAR, complement-dependent cytotoxicity assays. And then ICHE14 highlights cardiac safety, and the HERG assay has been used for a number of years. And more recently, the SIPA has been used to predict cardiovascular risk. And I think Bruce will talk a bit more about that one in detail, but that's another one that's been in development. So, and NAMs have also been used in development of a number of approved drugs. Currently, just in a little review of the literature, that NAMs have been used for development of Kalydeco for cystic fibrosis, where an in vitro chloride transport assay was used, which ultimately expanded the Kalydeco indication to patients with specific mutations. For Galafold, a enzymatic in vitro assay was used with HEK293 cells expressing mutant GLA genes. And this also expanded the indication for this product into patients with a defined mutation. And more recently, and I think more importantly, NAMs have been used pretty extensively and you'll see why in a minute, but for monoclonal antibodies. In the case of Veopause, in vitro assays for cytotoxicity and complement inhibition were used with the human proteins. And these confirmatory data helped support product efficacy and ultimately approval of the product. And then for Kimtrac, again, human cells and tissue-based assays were used since this product did not interact with or cross-react with human cells. And ultimately, using this approach, it demonstrated high affinity and binding for its human-specific target. So, as many of you are probably aware, FDA's initial focus is on NAMs for use in monoclonal antibody testing for monospecific, with one target, monoclonal antibodies. In April 2025, FDA released its overall roadmap, highlighting that it would focus on monoclonal antibodies as a first test case. And ultimately, in just a month ago, in December, FDA released its draft guidance on streamlined non-clinical safety testing for monoclonal antibodies. And the highlights of this, essentially, one can use lower species when it's appropriate and biologically relevant if the target is present in the species. And secondly, studies of greater than 3 months duration typically no longer required in large animals when supported by a weight of evidence approach. And in the absence of any pharmacologically relevant species, a weight of evidence approach may be used for non-clinical safety testing of monoclonals. So certainly, again, I'm getting close to the end here, but there's always advantages and disadvantages of using any new approach. There's a number of key advantages of using NAMs in your product development program, including the fact that these are human models. The data you obtain from them will be much more relevant than from any animal model. And certainly this drives use of fewer animals in the process. And the nice thing about NAMs is they can address very specific key questions. They're usually very targeted and leveraging human cells and tissues. And NAMs are supported by NIH and numerous regulatory bodies throughout the world. So there's certainly differences in what these different groups will accept. However, I think that'll be pretty well laid out in guidance documents, and there's going to be a lot of activity in the coming decade for sure. NAMs should save you money in program development and improve predictability of your safety program because you're using human models. But then again, there are certainly challenges with using NAMs, including the time it takes to develop and validate a novel NAM if one's not already available, the rigorous testing and validation that they have to go through. And there still will likely be some continued need over the next decade or so for certain animal studies. We're not ruling those out completely, but hopefully we'll be using fewer animals and obtaining better data with the human products over the next coming years. And it's not certain whether all regulatory bodies will accept a given NAM. While the FDA may accept it, it may not be accepted by the EMA or Japanese regulators, for instance. And there's certainly changing in data requirements. The things are evolving all the time, even as we present this webinar today. So how do you go about this, approaching the use of a novel NAM and incorporating it into your program? Well, briefly assess where in your program a NAM might be useful or needed, and then determine what type of NAM, what type of experiment will support your program. Is this NAM validated or qualified for use? If it's validated, then it can be used in agreement with regulators. If it's qualified, it can be used in tandem with other supporting approaches. So can you use it to justify its use in lieu of an animal study completely? So what I would do is propose your approach to the FDA at a meeting, such as a pre-IND meeting, how you want to leverage this NAM, and then obtain FDA feedback on the approach, your model, the endpoints you're testing, and the study scope. The overall plan to use how you will use this NAM to address these key questions, and then incorporate FDA's feedback into your NAM design strategy for submission to the regulatory authorities. And hopefully if the study goes well, the FDA should accept it and add to your program. So as I come to the end here, I just want to highlight a few key thoughts. Take a look at the FDA roadmap that came out in 2020, April of last year. Certainly, there's a lot of good points, highlights there, and how to reduce animal use. The FDA has provided specific guidance how to use NAMs for monoclonal antibodies, but NAMs can be used for just about any product. So consider them no matter what product you're developing, if it's relevant and accepted. But the NAMs field is certainly rapidly evolving. There's going to be new tests and models every day, and you have to be sure that they're accepted by the regulatory authorities. And in fact, this may take years to fully implement. We're just at the beginning of this process. So don't expect big changes overnight. Some people are expecting significant shifts, but it may still take 5 or 10 years before these are fully implemented. And finally, discuss the use of NAMs within your program in context with your consultant or other experts before talking with FDA to determine feasibility of using NAMs to support your non-clinical safety program. So I think that wraps it up for mine. I have a few references. If my slides advance. Yeah, so I have a few slides. I have a few references that will be included in the slide deck for your reference. And with that, I'll pass it over to my colleague, Bruce Pearce. Thank you very much.
Thank you, David. Hello, everyone. I'm Bruce Pearce. David's provided an excellent overview of NAMs and introduction and touched on a number of important aspects of NAMs and the use of NAMs in the regulatory environment. Talked a little bit about the incorporation of NAMs into regulatory documents that you're familiar with already, and talked a little bit about the regulatory strategy, how you approach FDA and talk about NAMs, what things are important. And I'd like to change gears and talk about the use of NAMs in regulatory decision making. And now I have control. Okay. As David mentioned, 2025 was an important year in terms of announcements from the FDA about NAMs. In fact, in April 10th, FDA announced that they're phasing out animal testing requirements for monoclonal antibodies and other drugs. And this sort of lit up the phones in terms of response of individuals requesting information and feedback about whether or not they can employ these approaches, not use animal models and accelerate their development programs. How can they do that? And one of the things that I did repeatedly was to draw their attention to FDA's roadmap that was introduced about the same time, indicating that this is a phased adoption program. And that on the near term, within the first 1 to 3 years, monoclonal antibodies, as David mentioned, are the pathfinder class on this roadmap. And on the medium term, within 3 to 5 years, that they'll expand NAMs to beyond monoclonal antibodies to include broader biologics and small molecules. And ultimately, in the long term, the goal is to make animal testing largely obsolete and replace it with a standardized NAM toolbox. In addition, David mentioned the fact that in December, just about a month ago, FDA released the draft guidance on reducing testing in non-human primates for monoclonal antibodies. And this caused another flurry of activity for those sponsors that are working with non-human primates or are thinking about the program going forward and should they consider a different approach. And again, I had to bring them back to the FDA roadmap and explain this. But the significance of this is that it represents the initial implementation of the FDA roadmap in terms of reducing animal testing for a particular class of drugs. At this point, I think we should all be thinking about animal testing as the exception rather than the rule in terms of looking forward. And we should also be thinking about establishing NAMs literacy as this transition from the initial use in monoclonal antibodies and expanded use, that we should all be paying attention to trying to develop our own particular toolbox of NAMs that we use with our particular drugs and advancing our understanding and literacy. And this will include which NAMs can be used today to effectively address sponsor needs for regulatory applications, understand the requirements for NAMs to be acceptable for regulatory decision-making, which we'll talk about a little, and understand the distinction between validation versus qualification. Clearly identify drug classes that are NAMs applicable and the use of NAMs in standalone capacity versus complementary use, where the emphasis is today, and resources for NAMs implementation. One of the things that I wanted to do as a part of today's presentation is to identify resources for you that you can turn to, particularly if you're new to this particular area, to begin to advance your NAMs literacy. In this slide, I have NAMs that are broadly recognized as acceptable for regulatory decision-making. David's talked about these, and I want to make several additional points. There are validated alternatives, which David noted have been around for a long time, have been used for quite a long time. Then there's weight of evidence for safety decisions. Now, we talk about weight of evidence in terms of the weight of evidence framework, and we can also speak about weight of evidence as an NAM when it's used to reduce the use of animals in particular testing. And the quintessential example of that is in carcinogenicity testing, which we'll come back to. Developmental and reproductive toxicity, weight of evidence is used here with NAMs to assess risk of malformations and embryo fetal lethality, and cardiac proarrhythmia prediction frameworks to look at the risk of serious cardiac events. This is often also involves the use of weight of evidence in conjunction with the SIPA assay. And then we have computational toxicology for mutagenic impurities, something that's been around for a long time. Many of you are familiar with this. One of the points I want to make here at the end is that most NAMs operate within the complementary context of use. That is, they involve the use of weight of evidence. They operate within the weight of evidence framework. And because of that, what I want to do is spend a little time talking about examples of the use of weight of evidence. And David mentioned that I would come back to the use of this in carcinogenicity testing. And this is a quintessential, I think, example of the use of weight of evidence to reduce the animal testing, the number of animals that are used in testing. And this is used with small molecules for carcinogenicity. So instead of conducting a 2-year rat study, sponsors can submit a weight of evidence assessment to prove that it's not necessary, or to provide strong evidence that it's not necessary. In terms of one, that the molecule, the test molecule is clearly carcinogenic, so there's no need to do, it's not warranted to do a 2-year study, or that it's very unlikely that it's carcinogenic. This is really important because the 2-year study, of course, it takes all of this time, but involves the use of a lot of animals and is very expensive. So this represents an important use of NAMs in the overall small molecule development pathway. This is also a very good example in terms of seeing how weight of evidence is used in regulatory decision-making, how it's structured, and the history of what it took to get to this point. I think one of the things you have to appreciate, NAMs don't arrive overnight. And in this particular case, the program was initiated, ICHS-1, in 2013 to examine, to collect data from sponsors to assess whether or not weight of evidence can be used to reduce testing. That is, was it predictive for that particular use? So, and the ICH S1B R1 guidance document was finalized in 2022. So that was about 9 years that was involved in taking it from initiation to introducing it into use. Another example of the use of weight of evidence for NAMs is in reproductive toxicity. And in particular, and David mentioned this, the use in the risk of malformations and embryo fetal lethality. And this involves using a battery of assays to assess that risk and weight of evidence to combine these data with in vivo study data, et cetera, to help to facilitate the introduction of women of childbearing age into phase 1 and phase 2 clinical trials. This is actually the use of this, the introduction and the opportunity to use a weight of evidence or to use weight of evidence plus NAMs in the ICH S5 R3 guidance is viewed as by some, that is, as a monumental change in terms of regulatory acceptance of this. However, even though this represents a monumental change in the position of regulators, companies really submit developmental NAMs to health authorities for qualification and the intent to support regulatory decision making. And this sort of highlights the fact that confidence needs to develop in NAMs for them to be used readily by sponsors. And there has to be confidence that the regulatory agents are going to view these and analyze these and make decisions in the same way that sponsors do. Carrie, going further with implementation of NAMs, I wanted to point out this particular website, the FDA website, that covers the agency-wide efforts for advancing development, qualification, and implementation of new alternative methods for product testing. This is an important resource for those of you that are new to this and that want to, or those that want to further their NAMS literacy. This is an excellent resource in terms of learning what's going on, what programs are underway at FDA, and on this website are listed publications related to NAMS, and some of them are co-authored by FDA. Now what I'd like to do when we're talking about validation and qualification of NAMS, distinguish between the concept of validation and qualification. And when we talk about NAMS, we usually talk about the qualification for NAMS, and that's important. Validation, I think many of you are familiar with the fact that it's the process of demonstrating the method's reliable and relevance for its intended technical purpose. It's independent of specific regulatory decision, whereas qualification is a regulatory determination that a method or a tool is acceptable for a specific regulatory use in a defined context of use. And this is really important, context of use when it comes to NAMs. The other point that I wanted to make on this slide is that we're in qualification in NAMs. We're turning our focus toward qualification in the human context. That is that the NAMs, the endpoints for NAMs reflect human biology. They're important to human biology, and they're predictive of outcomes in humans, particularly adverse outcomes. And so the biologic event that's targeted by the NAM has to relate to the adverse outcome pathway for NAMs. A number of key concepts for regulatory use of NAMs are outlined here. And David made reference to the validation and qualification and regulatory acceptance of new approach methodologies published by ICFAM in 2024. And this document describes these particularly important concepts in terms of thinking about the use of NAMs. That one, you have a well-defined context of use. This is sort of like a foundation statement in your regulatory submission and arguments for the use of the NAM supporting the regulatory decision. There should be clear demonstration of biological relevance to the clinical indication. Technical characterization, data integrity and validity of results, and information transparency. And I want to just talk a little bit more about the first two in this list, where the others, technical characterization, data integrity, and so on, are those concepts that are related to concepts that many of you are familiar with within the GLP environment. So in terms of context of use, I put together a generic statement here to give you an idea of what the components of a context of use statement will be. And this is important that this is included in your regulatory document to make it clear to regulatory reviewers that, in fact, you have a clear idea of how you represent a clear idea of how your NAM is being used. So this statement goes on that NAM will be used for a particular regulatory decision, for a drug or class of drugs for a particular indication by evaluating endpoints and a particular model system across exposure range, clinically relevant exposure range, and that the NAM is fit for purpose in the applicability domain and will be interpreted based on decision rules and thresholds prospectively defined and comparatives like positive and negative controls. In addition, it's important to identify that where a NAM will not be used, that you include what decision will be excluded. So you make it clear that you're staying in the context of use and that you point out what the limitations are. Understand that NAMs that you find that are being used to support regulatory decisions are qualified within the context of use. If you go outside the context of use, then this is going to represent problems if it's perceived that way by regulators. This is going to represent an issue for you in terms of getting regulatory reviewers to accept your NAM. Biological relevance. This is a key aspect of this. Like I said, we're shifting toward focus on biological relevance in humans. And what I've included here is a statement within the pro-arrhythmia risk, the SIPA paradigm. And the reason I've chosen this is it's an excellent representation of biological relevance. The NAM evaluates drug effects on human cardiac repolarization by measuring integrated multi-ion channel activity, action potential duration in human cardiomyocytes. This biological process directly underlie torsade-of risk, which is dose-limiting safety liability for systemically administered therapies in the intended indication. So how does that statement support biological relevance? Well, the clinical hazard arises from the disrupted cardiac repolarization. The NAM measures the same mechanistic drivers for this pattern, and the endpoints measured in the NAM directly translate to clinical ECG findings. So this is like the ideal example, and FDA has publicly stated that SIPA is biologically relevant for human pro-arrhythmia risk assessment. So this shows you an example of the kind of connection that you need to make between the measurements, the NAM that you're using, and the clinical question that's being addressed and is important to regulatory decision making. I'd like to turn now to talk about the validation and qualification network, and to point out that this is important. This is another resource for you. The validation and quantification network is a part of the complement RE program. This is sponsored by the NIH in collaboration with FDA and many, many stakeholders. And I wanted to make a point. This represents infrastructure that's being set up recently to help to support the acceleration of the acceptance and development of NAMs that can be used for regulatory decision making. The other point I want to make about it, again, is that NAMs don't just materialize overnight. It takes a long time. And this program shows that that this began in 2025, and it shows implementations in 31 and 35. So analogous to FDA's 5-year program, the introduction of candidate NAM is envisioning that it will take at least 5 years for this NAM to be accepted. So 2025 marks a regulatory inflection point. Policy direction is explicit. Initial implementation focuses on monoclonal antibodies. FDA is supporting this validation qualification handbook, if you will, and the infrastructure to support adoption of NAMs is beginning to develop. Will be there and to help to accelerate this. And I have a diagram on the right-hand side. I did a PubMed search to look at the number of publications per year, and you can see that that relate to NAMs. And you can see 2025 does indeed, based on these data, represent an inflection point. And sort of final thoughts. Alternative methods for regulatory purposes began back in the 1970s with mutagenicity and in vitro screening assays that matured formally validated in regulatory replacements in the 1990s and have evolved into mechanistic decision critical NAMs in the past decade. FDA has incorporated validated alternatives to in vivo non-human tests in general recommendations for non-clinical drug development and is accepting NAMs for regulatory decision-making when scientifically justified and appropriate. And that's an important qualifier. As you can see, that really takes a lot to get to that point. FDA is also encouraging sponsors to submit non-clinical test NAMs for regulatory review in parallel with animal studies. However, this is voluntary. It's not a requirement. That word should be dropped, but it's voluntary. So where are we now? I mean, the idea of one of the themes here in this presentation is to help to clear away the noise and help you to understand where we are with this, to give you a 30,000-foot view on this. And I think that this is best represented by a quote from a paper published last year by FDA, CDER, and Office of New Drugs, entitled Experience with New Approach Methodologies. And this quote that I've extracted from that publication is, there's still need in most cases for general repeat dose non-human toxicity studies to assess the risks and the safety of many new drugs. And we're at the beginning of this transition from the initial reduction of animal testing to realizing the NAMS toolbox. And I hope that our snapshots that we presented today with regard to the use of NAMs, the requirements for them, some of the most important properties, and we've provided resources to help you to advance your NAMS literacy. Thank you very much.
Thank you so much, Bruce and David, for that fascinating conversation. I'd like to turn it over to the Q&A for today's webcast. And the question we have here in the chat is, often biologics have ADAs, and that drives the sponsor to consider NAMs to waive the greater than 3-month toxicity study. In your experience, should a pre-IND meeting be used, or after a submission of the IND, go for a type C?
Well, I'll just chime in briefly, Anna. Thanks. I think the pre-IND meeting, you don't know what the results are going to be necessarily. Hopefully you get your feedback from the agency at the pre-IND meeting on the design of your GLP safety study. Then subsequently, you may, whether you see those ADAs or not, it may be something you do later on. Earlier the better generally, but you don't know whether you're going to get that input. You could ask the question at the pre-IND stage, but it may be better to wait to see sort of how the animals, at least the 1-month study, would turn out. I don't know, Bruce, do you have any specific experience or thoughts?
Yeah, I agree with what you said, Dave. I think that if you have already done dose range finding studies and some repeat dose toxicity studies, you'll have seen the development of ADAs to your drug. And if they're neutralizing and they're preventing the biologic action of the drug, then I think that in the pre-IND meeting, or even you might consider an interact meeting, because this is a significant problem in terms of using, deciding on alternative species or whether or not you're going to use NAMs. So I think that might be a really good setting to take this up in. Of course, like David said, if you don't know until after you've had the IND meeting, probably a Type C meeting would be a great idea. If you're going to make a change in a program, it's always a good idea to discuss this with FDA and get their feedback. Not only will you get feedback, but you're going to get some advice for FDA, helpful advice in terms of what direction to go in.
Excellent. And that turns us to the next question. If you had to identify one thing that is most important to whether FDA will accept a particular NAM, what would that be?
Good question. I think that maybe the one thing is not having a clear statement of context of use. As I said, that the qualification of the NAM is within that context of use. And if you don't clearly define what that is and demonstrate that you're limiting the use of the NAM in terms of supporting the regulatory decision to those boundaries originally established for that NAM, the context of use. So I think that this is something we'll derail it. In terms of your approach, overall approach, leading with the technology that you have, the assay that you have, and not thinking about the regulatory decision. It's, being focused on the technology and the sophistication of the assays that you're using and not thinking about whether or not they actually are clinically relevant, they are biologically relevant and directly impact the outcome, the adverse outcome in humans. I think will lead you down the wrong path.
Excellent. I think we may have time for one more. What are some of the reasons that NAMs are not accepted?
Yeah, I'll just chime in quickly and give Bruce a chance to give his 2 cents. But certainly just, robustness, non-relevance to the human situation. They really have to be, validation certainly is if they can survive that process. And human relevance is key, and showing that, they mimic the biological process or the expected process clinically. And hopefully they do, because they're ideally human. Extrapolating data from an in vitro model to human, a full in vivo human situation is always challenging. But I think having a adequately specific, focused NAM that addresses a key question, like a few of the examples I presented for the approved products, where they really are looking at very key questions to expand an indication or something like that, trying to make them too broad or all-encompassing could result in NAM's failure.
Excellent. I do see one more question here, and we do have 2 minutes left, so I just want to make sure this one gets answered. What do you see as the biggest regulatory challenge today in broader adoption of NAMs?
Give it a go, Bruce.
Mm-hmm. Yeah, I think the concern is, and the example of this was in reproductive toxicology. Despite the fact that the guidance document indicates that it's acceptable to pursue NAMs, the fact of the matter is that the industry is reluctant to do that. They're reluctant because they're not confident that in this complex environment of reproductive toxicology, where this dynamic system that you're assessing in terms of safety, that your understanding of how to interpret the NAM and how to use it is different from what the regulator has. That's, I think, from the perspective of the sponsor, that could be an issue. From the point of view of regulators, introducing new NAMs, this is something that you do in coordination with FDA and with NIH, perhaps, to develop a NAM, because it's a long path to establish that and to qualify the NAM for use within the context of use, and as David said, target their clinically relevant outcome. I hope that answers the question.
Excellent. Thank you very much, Bruce. I think we're just about at time now, so I'd like to take a moment to thank our panelists very much for their insight and their information, as well as all of the folks and the attendees who have provided us with questions and joined today's webcast. And very much appreciate you joining, and thank you for the lively discussion and the very interesting topic. And that concludes our webcast.
Yeah, thank you all.
Thank you very much.